just say I've been waiting 10 years for this conference, so Marcia, thank you. Sex mismatch, gender, immunology, and even pregnancy, right? Oh man, I'm all over this. This is great. It's kind of funny. I used to study yeast and chaperone-mediated protein folding, and then I said I need to study systems biology. So I'm a translator, and my introduction slide. This is my overview. This is the outline as well. So on the left, you see how we do bone marrow transplants. It's very schematic. You take a graft from the donor, you put it in the recipient, and you're really interested in can you cure the cancer? That's what the patient wants to know. But of course, the biological phenomena of reconstituting an immune system in a new host can never really be fully understood or completely studied. And we have tremendous tools here. We do 230 allo transplants a year at Stanford. We bank blood for peripheral blood mononuclear cells and plasma and DNA content. We collect prospective samples of all those patients at 30, 60, 90, 180, 270, 300 a year and a half out to 3 years, 10 samples per patient. They're all 95% viable, all liquid nitrogen, tremendous samples. And then, of course, you have pregnancy. And pregnancy is another tremendous experiment. Everybody expects a normal pregnancy, right? So all the people that have problems with pregnancy show up to be studied. And, it's amazing that you'll hear my story. I had to go to Copenhagen to find my colleagues to work with, but probably because I didn't look enough around here. So I'm hoping there's some in the room, and we'll speak. So that's my overview. And then the other part of this whole story is simply, if you think about biology, right? I was a yeast chaperone and a mediated protein folding person. And I had to go somewhere else. I mean, I really had to get into the disease, the biology. And so you have to find signals that you can actually measure. So here's a simple experiment for whom we should blame Irv Weissman. So Irv is not in the room, but I would tell it the exact same way. He's the third author on that paper on the left from 1955. He was 18 years old in high school chasing field mice through the western hills of Montana. And the experiment is very simple. They did skin transplants, right? Take skin off one, put it onto the other, and they did gender combinations. And so you quickly see that it's the male skin onto the female that results in, where is it now? Male onto female, that results in rejection. And this, of course, is obvious now. We think about it. It's 1955. It was strain non-specific. Take it off of the male, put it onto the female, and it turns ugly. The fur falls off, and it falls away. And in fact, you can make it happen much more quickly if you take T cells from a primed animal, put it into the mouse, and now the skin will fall off. And you can show that it's actually T cells by separating those cell processes. In fact, you can now do a transgenic T cell experiment where you just put one T cell clone in that sees a molecule for either UTY or SMC, one of the 9 H-Y antigens I'll talk about today, and create the same exact skin rejection phenomena. Now, in bone marrow transplantation, that's my field, that's what I do 2 days a week, 3 months of the year. It's a phenomenally interesting sex mismatch problem. If you're a male patient and you have a brother, then let's just set the bar for how much graft versus host disease, that is the detrimental immune response from the donor against normal tissues, the skin, the gut, the liver, that we want to preserve, versus curing the cancer and having the patient not relapse their cancer. And the benefit of an allogeneic transplant is only evident when you have a not identical sibling. If you do this with syngeneic twins, it doesn't work. So clearly the donor's immune system is seeing the differences. And if there's too much difference, for example, MHC or HLA being mismatched, then you end up with overwhelming graft versus host disease and death. But if you can match for the major effects and then leave the minor effects free to play, then you can end up with a phenomenon where you'll not have too much graft versus host disease and not have disease come back. But now if we compare the female donor versus the male donor, the sister versus the brother, what's obvious and what I'm trying to make the point of is there's 40% more graft versus host disease if you choose the female donor than if you choose the brother, and there's 35% less disease relapse. So it's the yin and the yang, the good and the bad, the green and the red. It's what we deal with all the time. And it was smacking me in the face back in 1998 when I said, I want to study how we could monitor for success in bone marrow transplant. How could we monitor a blood test for an immune response that's going to tell us that the patient is not going to relapse their cancer and is not going to develop graft versus host disease? And so there was at that time, it was a good time to take this study on because the Human Genome Project, the first publication was David Page's publication in 1998 of the Y chromosome. And so when they published it, there was a whole bunch of the Y chromosome that is non-functional or exactly identical to the X chromosome. But then there were a series, and actually 9 genes that are highly expressed, not on the X chromosome, not X inactivated. And the X and the Ys are 91 to 99% identical. One of the molecules, I'm not even sure I see it on here. Yeah, elongation 1AY, it's 150 amino acids. There's 1 amino acid difference. And that amino acid is recognized in 10% of my males with female donors. And other molecules, like my favorite, DDX3Y, there's about 60 amino acid differences in a 650 amino acid. So this is a tremendously immunogenic, potentially immunogenic cadre cohort of molecules. And if you thought about the other problem, right, if you wanted to study bone marrow transplantation, you just said, well, I'm just going to randomly start sequencing everything. Well, there's 25,000 genes. The SNP rates are what, 5 to 15% allele frequencies, and so if you randomly choose male, female, or donor recipient, or brother, sister, or brother, brother, you'll be doing a lot of gene sequencing before you actually get to study the immune responses for a disparate allele. Whereas here is a situation where 25% of our donors are female, and they go into the male recipients. And in fact, when the problems develop, it's 40% of the male with female donors who have the chronic graft-versus-host disease. So I have no shortage of patients to study, and I have a very limited collection of target proteins I need to study to get answers. And people all want to know, what did the proteins do? And I honestly don't care. But since I get asked these questions all the time, I have to say, well, they have fascinating roles. I don't really know what they do. They do zinc finger binding proteins. David Page has been studying ZFY for about 25 years. I don't think he still knows what it does. UTY and SMCY are certainly the most fascinating molecules because through demethylation and epigenetic effects, they probably are the molecules that make us male. And I'll just leave it there and tell you to go see some of Howard Chang's work and other people's work. But the truth is, I study them because of their targets, right? Because they're disparate. 95% identical gives us 5% differences. And in 1998, there was a history of T-cell immunity, right, Mark? Because T-cells are sexy, and everybody wants to study T-cells. Nobody studied B-cells in 1998. So the phenomena of the skin transplant was recognized in 1955, and Els Gulmi, who's been very supportive of my work, had shown as early as 77 that you could have T-cell responses that are HLA restricted in the female mounting against the male. And she goes on to have a cottage industry in discovering HY epitopes in male with female donors. Finally, in 1995, the biochemical isolation of the first small 10 amino acid peptide that is representative of the minor histocompatibility antigen, which remains our actually most dominant T cell response. It's a protein called SMCY, and it is still the dominant molecule. And then tetramers, a la Mark Davis technology, allow us to quantify those T cells in blood and to try to make clinical correlations. They were very unimpressive correlations being made following T cells. And so along comes my interest in 98, and I said, well, this is fascinating. T cells see a minor histocompatibility antigen here represented as a red small dot, which is, of course, part of the proteasome in the cytosol, where you chew up all cytosolic proteins, and some of them fit into HLA and are then going to be capable of being recognized by a polymorphic receptor. Highly schematized picture here. And then on the other hand is the globule with an antibody. We all know what an antibody looks like. And what was the advantage of antibody in this analysis? Well, the repertoire is actually more complex. It goes on actually happens to be what I really study now is antibody complex repertoires, and that you could find single amino acid differences that are recognized by these antibody responses in male and female donors. I told you about one already. And most importantly, there's no cell culture. The antibodies are stable in the blood and the system in the patient's freezer, and I could do this work not working on weekends. So this was perfect. So it's important to know why you do things. So I made 5 recombinant proteins and their X homologs, and these were made in a number of animal systems. Turned out to be E. coli on a nickel his affinity expression system was perfect. And as I said, some of the molecules have 1 amino acid difference, and some have many more. Some of the molecules are quite large, and some of them are very small, and it doesn't really matter. You make these molecules, you throw them up onto an ELISA plate, and then you start asking, do people make antibodies against this? And of course, there's years worth of work trying to optimize how you measure antibodies in a reproducible fashion with an assay that you're going to start making clinical decisions based on. But this is a heat map from a paper in 2005 where we show 75 male with female donors being tested against 10 different proteins. On the left panel are the Y antigens, my favorite DBY. Names aren't important. Different proteins. And then on the right panel, they're X homologs. Again, they're 95% identical. So I show this graph to make a couple points. One is these are the male with female donors, and if I showed you the normal males, they'd all be blue. Okay, so males don't have antibodies. In fact, that's how we develop the assay. We take the normal male serum and we decide that since they should be serologically negative, let's measure 150 of them and let's allow a couple outliers. But essentially, the threshold is based on normal male serum. And then if you look at a male with female after transplant, the response against X, which should be self in the system, when we see a self, we also see a stronger Y. So anytime we see an X, we see a stronger Y. And it turns out over years of work, this is 2005, we now know that the initial immune response in patients after transplant, where the male patients are always negative before transplant, have no antibody responses here showing UTY and DBY, no antibody responses for the first 2 months. Around 3 months start to develop antibodies that then plateau and they remain elevated for 10 plus years post-transplant. So we have a convalescent serum, it remains seropositive. And those of you who are EBV positive, you understand the concept of serum remaining positive for life. Now, the X and Y thing is phenomenally interesting. We don't have time to talk about it, of course, but it's the initial non-specific response, which then becomes very specific. The X goes away, the Y becomes very tight, very high avidity, and that's also associated with a transition from IgM to IgG. But what was fascinating was if we took those 75 males and said, who's seropositive, shown with the hatch marks, with the solid lines, and chronic graft-versus-host disease is show you in a second, the detrimental problem that develops on average 9 months after transplant in about 50% of your patients. And relapse, well, relapse is easy. That's when cancer comes back after the transplant. So patients who are seropositive in blue have a high frequency of chronic GVHD, 91%. That is really tremendously predictive, ridiculous p-values. And none of the patients relapsed who were seropositive in this cadre after 6 months post-transplant. And then in between are the patients who are seronegative, and you can say, well, there's 25,000 other molecules that are potentially eliciting immune responses causing chronic GVHD. But we sure captured a lot of signal, didn't we, for 4 things. This is what chronic GVHD can look like. And it is dramatic. It's the booby prize of allogeneic transplantation. Transplant's been going on since 1968. We certainly had more of it than we have now. But it is got features of essentially any autoimmune disease you want to describe. Scleroderma is the first that jumps to mind. Lots of fasciitis, skin changes, hide-bound skin changes, oral pharyngeal changes, dry eyes, ducts and problems with making enough fluids, whether it's tears or saliva. And pulmonary fibrosis can be the problem which kills the patients. It's poorly understood. It happens in half the patients, and it's really a steroid requiring therapy, and we need better treatments, and this is what we focus a lot of our efforts on, especially with the shown association of H-Y antibodies. So it was a good time to have that. And I just want to go on and say now, that was a cross-sectional picture of patients' captured serums at all different times after transplant. This is a prospective analysis. And what I can convince you of right away, just showing an example of UTY, just to make this simple, nobody has an antibody at 2 months. Antibodies develop at 3 months. Antibodies usually persist. And antibodies come on in association here in a quantifiable manner with higher amounts of antibody associated with higher risk of chronic GVHD. And we can score against multiple antigens, and anybody who has multiple antigen responses is going to have essentially 100% chance of developing this chronic GVHD problem. So this is what you do with assays, right? Ultimately, assays become useful for you. They become useful because they predict an outcome, or they become useful because they let you understand the underlying pathogenesis of what might be happening. So the association of B-cell alloimmunity suggested that there could be a critical and targetable role for B cells in the treatment or prevention of chronic GVHD, and this is what I call the Rituximab hypothesis. Rituximab targets CD20, a B-cell antigen expressed about midway through B-cell development. B cells develop in the bone marrow from common lymphoid progenitors, hematopoietic hematopoietic stem cells undergo complex VDJ T-cell rearrangement, B cell rearrangements. And in that heavy chain, light chain rearrangement, once they can make a effective antibody or B cell response, then they can escape the bone marrow space. They have a new name, translational B cell. They're looking for antigens to interact with. Some of that interaction occurs in germinal centers, and ultimately they want to go back home, live in the bone marrow again as a plasma cell making antibodies. So you can see the unique point about CD20 expression. It affects mostly cells that are looking for antigens that are out in the body and not in the tucked away bone marrow space. And if you ablated those cells that express CD20, you would still have a source to make new B cells from the common lymphoid and progenitor B cells, and you would protect your long-lived immune responses against infectious antigens, allowing your patients after transplant to survive. And so we did a lot of studies on rituximab over the last 10 years, and I'm not going to show you them too much. I'm simply going to say that it actually was immediately rewarding because we could show that treating with rituximab was allowing us to, again, eliminate the alloreactive antibody responses while maintaining protective immune responses as we suggested by our hypothesis. And this is a treatment of people with steroid, with new onset chronic GVHD, but we could have also shown you long-lived plasma cell failures, that is patients who've had chronic GVHD for years, and obviously much of those B cell responses have become plasma cells, and they would not respond to these types of an anti-CD20 therapy. What is most probably effective about rituximab is prophylaxis. It's not the most potent drug in the world. You can kill a few logs of B cells, and that is good if you haven't already started the vaccine response. If you try to stop a vaccine with rituximab, it doesn't work. So it's not my intent to come on and tell you everything I've done for the last 10 years, but it's my intent to show you how you can start with a hypothesis, get some insights into its underlying pathogenesis, and then use your model system to monitor how effective you're being. Here's data for a new onset chronic GVHD treatment trial where patients have lots of CD19 positive B cells. You give a rituximab anti-CD20 depletion, and then 2 and 6 months later you have no measurable B cells. It's not surprising that we can show you, as I did on the previous slide, that the allogeneic antibody responses go away. But what do you think happens as the B cells recover here? Either we eliminated those alloreactive B cells, and these are now from the common lymphoid progenitor stem cells and are going to be non-responding in this new host environment, or we did a lousy job with rituximab and only killed a couple logs and now they're coming back. Well, it won't surprise you that first attempts never are as good as you hope for. So we've developed tools where we've gone beyond the antibody, and now using large protein microarrays to screen for small epitopes of peptides that are overlapping across the entire surface of these 9 H-Y antigens, we have found immune-dominant epitopes. This DBY2 molecule is a small peptide, 18 amino acids, that differs at 8 amino acids in comparison with its X-homolog. And so we can biotinylate it, put a fluorescent molecule on it, and much like a T-cell tetramer, start to stain B cells. We use a little trick staining for the amount of B cell receptor on the surface by staining for lambda at the same time. And it's highly schematized data again, but makes the simple point. 15 of these patients who were on the trial were male with female, and 9 of them had B cells and antibodies going into the Rituxan treatment. The B cells went away, and after the treatment, they all came back. And so did the antibodies, and so does the chronic GVHD. So you get a response for a while, and then you fail. And don't worry, it's good news. It gave us insights into what to do next. And so fortunately for us, a new drug called ibrutinib, it's a B cell receptor Burton tyrosine kinase molecule, has been FDA approved for the treatment of mantle cell and CLL, and we already have data showing incredible responses in patients with chronic GVHD. And the company is so excited about this that Mark will never believe this. A company-sponsored phase 1 study, never been done before in bone marrow transplantation, or I would say in autoimmunity, usually relying on investigators to bring the crazy ideas forward and let us take the first risks. So the company is so excited because of data like this and some animal model data showing very similar results that they're interested in going forward. So that's the first part of my story. That's all about transplantation. So, what was the lesson? Well, in our model system, we use sex-mismatch, right? So we brutally associated the Y antigen disparity as the target molecule. And then the second aspect of this was it possible that the female immune system was more robust than the male? I guess it was. wasn't fully worked out in my system, right? We could wait for Holden to try to figure that out. So for me, it was really the disparity of the immunological differences in the male with the female donors. The other point of this whole thing, though, was I want to reemphasize again that once you start to have antigen-specific targets for your hypotheses, then you can actually work more towards pathogenesis and therapeutic benefits. It's very hard to see that guy with scruffed up skin, hide-bound skin, ulcers, and other changes that probably are irreversible. How's he ever going to show me a clinical response? And yet he wants a clinical benefit, and he wants drugs that will benefit him over years. So we need to have correlative science. Okay, so my summary from that is HY is important, but how about this one? All right, so I was minding my own business about 6 years ago, and a woman from Copenhagen calls me up and says, I've been working with Allogeneic trying to find T cells in women who have problems with pregnancy loss, and I can't find a damn HY T cell, despite what they all say. And I said, well, I'm not surprised. And she said, would you mind looking for B cells or antibodies with me? I said, come on over. And so Henriette Nielsen is kind of one of those collaborations that just you dream about, okay? She writes all these papers, and everything I'm going to show you going forward here is really Henriette. And she came over and she did all the assays herself in my lab with me for a summer, and we had a grand old time. So there are people here who worry about pregnancy. 15% of pregnancy results in fetal wastage, and it increases as you get older. And there is a basic phenomena that what you've had previously does impact on what your children who follow are going to be like. This is a simple table showing that if you had prior brothers, then your birth weight is likely to be markedly reduced compared to those who did not. And if you have more brothers, you have more reduction than if you had females. And again, likely to have collaborators who do all the groundwork for you before they show up. And then she also shows that in nationalized healthcare states, you can do wonderful studies. She has a cohort of 500,000 women undergoing, who are in the birth ages of their life. And anybody who has problems with miscarriages has to come to her center in Copenhagen. And so she has no problem collecting 75 women who've had a normal first pregnancy, 3 lost pregnancies, and in the 5th pregnancy show up to get IVIG at 10 weeks post implantation with blood drawn and samples going forward. And so she says, I got 75 of these samples that bring them over. And so what she had shown was that if you looked backwards at those 75 women, and that's what we call secondary recurrent miscarriages, because there had been a first normal pregnancy with a healthy, and then there was subsequent, is that the easy button? And then there were subsequent normal pregnancies. Okay, and so what they showed was that a couple things. It's always a boy in the first pregnancy. And if you have a boy in the first pregnancy, then your chances of carrying to birth at 10 weeks in that 5th pregnancy is worse than if you had a girl in that first pregnancy, no matter what you do as an interventionalist. So they give IVIG, they do all kinds of monitoring, but if you had a brother, you're in trouble. And then the other issue here, and I'm not going to go too much into this, but basically they did all kinds of comparisons for whether the male effect was before or after the pregnancy of interest, and it was the pregnancy that was before. So the first pregnancy has the strong statistical effects. And so we came up with this HY hypothesis. It wasn't that difficult, was it? And we basically thought that the women were experiencing something against Y, and we wanted to test this. And so she brought her serum, and she brought me 75 controls, women who had 5 normal pregnancies. She brought the males, and she brought everything with her. And so basically I'm going to quickly just say that the females who were having these secondary recurrent miscarriages had multiple high antibody responses. And it was interesting because they had both Y and X. They really didn't have just Y alone. And this is markedly different than the control patients or the normal males. And how different was it? Well, as I said, there were 75, and they went on to have 44 that were seronegative, 33 that were seropositive. And the first question was, would they have a different outcome? Because we didn't know if they were going to actually have the same birth rate or not. And in fact, they have the same birth rate. And I was like, oh, well, I guess we're going back down to like some useless journal that never is going to be seen again. And then she said, oh, no, all they had were daughters. So the number of boys versus girls, it turns out there's a very well-accepted statistical power of it's 50-50 in the population. And so when you show a skewing where only most of the pregnancies resulted in females, we had something going there. So this is my summary for this study. Really, it was the data most suggests that it prevents implantation much more than the ongoing collection of successful pregnancy to term. And again, another example of sensitization. But why would I care about this? Well, anybody hear of lupus? So lupus women have children. When they have children, 2/3 daughters. Lupus women have sisters mostly. You can follow lupus effects on pregnancy going 3 generations. It's a gigantic effect. So was it the dysregulation that we're reading out as lupus, and that's what the pregnancy effect was? We don't know. We have grants in to try to study this, and Eliza I never get Eliza's name right, Chajasri, who was here at Stanford, is now at Oklahoma, has a huge cohort of patients, and we'll study that. So I'm going to stop. I'm going to say that there, of course, is always a need for collaboration in lots of people. And what I showed you today was work from Fong Wu, who does fantastic H-Y antibody arrays. Students, Christine's been with us, student Simmer, now a Stanford medical student, and Hideki came from Japan and really analyzed all this data. Thank you. Any questions? It was perfectly clear. I love it. Oh, sorry.
Well, there is another explanation for the male birth issue between SLE and that's also consistent with XY intragenomic competition, in which there's this imbalance between multi-copied genes with an X chromosome and the Y chromosome that skews the sex ratio, and so you could lose male births or male concepti because they develop abnormally as a consequence of XY intragenic competition.
How would I measure that immunologically?
It's not an immunological readout per se, but it's a genetic readout.
No, I care about it. Thank you. Thank you.